Hello learners, still is a remark for the second part of the video lecture on our session this week. So we're done with the discussion on the preparatory phase or the interphase. Now let's go to the phase where the cell divides. Okay, so we are going to talk specifically on mitosis. So mitosis is again also called as the equational division because the chromosomes are equally distributed to the daughter cells. Again, daughter cells are what are produced after mitosis or cell division. This means that the daughter cells produced after mitosis have the same amount of genetic materials. Also, the chromosome number of the cells is the same, or of the daughter cells, is the same with the chromosome number of the parent cell. And again, mitosis has four phases, which might sound familiar to you. These are the prophase, metaphase, anaphase, and telophase, which happen in sequence or in order. And to easily memorize the following, just remember PMAT or PMAT. PMAT, P for prophase, M for metaphase, A for anaphase, and T for telophase. Okay? Since these happen in sequence, let us start first with the discussion on prophase. In prophase, the chromatins start to condense or to become thicker. So from this state, they become thicker or they start to condense to become chromosomes, which are now visible under the microscope. Aside from that one, the nuclear envelope or the nuclear membrane, which is not shown in the figure, starts to disappear. Again, a nucleus has its own membrane, which is called as the nuclear envelope or nuclear membrane, and that is what is going to dissociate or disappear in this phase. Also, in animal cells, there are these special structures called as the centrioles. So these are the centrioles, and these centrioles are going to move to the opposite poles of the cell. Additionally, spindle fibers, which are made up of microtubules, and again, microtubules, based on the previous discussions, are one of the cytoskeleton components of the cell. And these microtubules start to form and start to attach to the kinetochores of the chromosome. Okay? And again, you've already learned about where kinetochores are found in the previous video. Okay, next. In metaphase, the chromosomes line up at the metaphase or mitotic plate or the central region of the cell. So, also, the attachment of the spindle fibers to the kinetochores to each chromatid is completed in this phase. So each chromatid now has an attached spindle fiber in their kinetochore. Okay? So that is what happens in M phase. So middle, they align, the M there stands for middle, they align in the middle portion or the middle plane of the cell. Next, after M phase is this one, anaphase. So this is where the centromeres or the central region of the chromosomes break apart, making now the sister chromatids separated into individual chromatids. So from sister chromatids, they are now going to split into individual chromatids. And these individual chromatids are going to be pulled away from each other by the spindle fibers by the help of centrioles to the opposite poles of the cell. Remember, anaphase A, they are being pulled away. Again, the chromatids are pulled away toward the opposite poles. And then lastly, the chromosomes are now found at the opposite poles. So this means that there must be two clusters of chromosomes found in the cell. In this phase, the nuclear envelope or membrane that disappeared during the prophase reassemble, and the chromosomes start to decondense. So this is now the a photo or another figure of that one. So this is the nuclear envelope or nuclear membrane that is starting to reappear. Okay, so that is what happens in telophase. So that's the end actually of mitosis. Now, sir, where is the division? Okay, in general, what actually happens in the M phase or the mitotic phase is just a karyokinesis or the division of the nucleus. So the cell actually did not divide in mitosis, but only the nucleus. So as seen in previous slides, again, what actually divided was just the nucleus. If that's the case, sir, where does the cell actually divide? The cells literally divide after telophase, which is not part of mitosis because again mitosis only has four parts: prophase, metaphase, anaphase, in which this part where the actual cell division happens is what we call as the cytokinesis. Now, in cytokinesis, this is where the cytoplasm or the cell splits into two or into half. From the word cyto, it means cell, and kinesis means move. So basically, this means that the portions of the cell move away from each other. And depending on the type of cell that is undergoing cytokinesis, mechanisms differ. For animals, so as what you can see there on the screen, there is a formation of a cleavage furrow. So this is the cleavage furrow. Since the outer layer of an animal cell is only made up of the plasma membrane, in which a plasma membrane appears to be fluid in manner, the plasma membrane is pinched inward by the contractile rings of microfilament. So these now are the contractile rings of microfilament that pinch the animal cell inwards, specifically in its central portion. Okay, this now forms two daughter cells after. So this pinching of the microfilaments create a cleavage. So the pinching now creates a cleavage. Hence the name cleavage furrow. Okay. So after pinching, there is now a formation of two daughter cells. Okay. On the other hand, plants have a different mechanism. Since plants have an additional outer layer called as the cell wall, which is of course hard, the forming of cleavage is not possible. Instead, there is a formation of what we call as the cell plate in the middle portion of the dividing cell. This cell plate is made up of vesicles. So these now are the vesicles. So this is the cell in cytokinesis, and these are the vesicles that are forming in the middle portion or in the middle plane of the cell. And these vesicles now, since they are also made up of the same components with the plasma membrane and of the cell wall, they fuse together. So in the second figure, these are the vesicles. They are going to fuse together, and afterwards, this portion now will merge into the plasma membrane of the plant cell and eventually with the cell wall of the plant cell, forming now two daughter cells after. Okay? In summary, actually that's the end of cytokinesis. So there is this is where the actual division of the cell happens. In summary, to better understand the process of mitosis, I have here an animation of mitosis. I got it from the Amoeba Sisters on YouTube. And again, mitosis has four phases, which are the PMAT, PMAT, wherein P stands for the prophase, M for metaphase, A for anaphase, and T for telophase. Okay? In prophase, this is what happens. From chromatin strands, they become compact or they condense together to form chromosomes, and these chromosomes now are visible under the microscope. Aside from that one, the nuclear envelope or nuclear membrane starts to disintegrate or dissociate. And also, still in prophase, the centrioles of animal cells start to migrate into the opposite poles of the cell. And lastly, the spindle fibers start to form, and also spindle fibers start to connect or to attach to the kinetochores of the individual chromatids. Okay? After prophase is the metaphase, wherein the chromosomes line up at the metaphase plate or the mitotic plate. And of course, the attachment of spindle fibers to the individual chromatids of the chromosomes is ended in this phase. And then after metaphase comes next is anaphase, wherein the individual chromatids are pulled away from each other. So from sister chromatids, the individual chromatids are going to be pulled away toward the opposite pole where the centrioles are found. Okay, just remember A for away. And then lastly, after the anaphase is the telophase. In telophase, this is what happens. So in telophase, the nuclear envelope or nuclear membrane, actually there are two, since there are two clusters of chromosomes now in the cell, so two nuclear membrane or envelope will start to reappear. And aside from that one, which is not shown there on the animation, the chromosome will start to decondense. Then of course, so that's the end of mitosis. It did not really divide the cell. The actual division of the cell actually happens in the next stage, which is called as the cytokinesis, which differs in division in plants and animals. In this case, this is a division in animals. So in animals, there is a formation of cleavage, and then a cleavage furrow, and on plants, there is a formation of the cell plate. So just remember that in cytokinesis, the cell splits into two. Okay, just in this case, so the cell splits into two. Okay, to sum it up, mitosis only happens in our body cells or somatic cells. Therefore, mitosis is responsible for forming our body cells. That is why we grow or we increase in size and our wounds heal. Thanks, mitosis. At the end of one division of one cell, there is a formation of two daughter cells. And these two daughter cells can continue to process to generate or to create or to produce more and more cells. That's why we grow. That's why the wounded or the abraded cells are being replaced because of this process, cell division, specifically mitosis. And again, in one parent cell, once it divides, it produces two daughter cells. And these two daughter cells are twins. Not just twins, but identical twins, because again, they have the same composition and the same number of chromosomes. And that is also the reason why mitosis is also called as the equational division. I guess that's the overall discussion on the mitosis, on the concepts of mitosis. So to give you a glimpse of how mitosis happens in actual cells, I have here micrographs of the onion cells undergoing mitosis. So on the left side, that's the prophase. So the cell, the chromosomes are visible. And aside from that one, nuclear envelope starts to disappear. And also next is the metaphase, wherein the chromosomes are now on the central portion of the cell. And then in anaphase, the individual chromatids are being pulled toward the opposite pole. And on the telophase, there is now the reformation of the nuclear envelope.